Biodiversity Conservation Foundation is an NGO based on Tamil Nadu with the vision to visualize a society for promoting and assisting in healthy coexistence of human and wildlife and ultimately ensuring conservation of biodiversity. Strategic implementation of this mission drove us to handle research and reaching stakeholders in a very synchronized manner. Our research extends from ants to elephants and exploring effective management practices in the perspective of biodiversity conservation. With our wide research team, we were able to work from Nagapatnam to Valley of Flowers, stretching across different landscapes and different people with varied dependency on the ecosystem. In a nutshell, we connect nature with people and people with nature. So let's get connected today also in the virtual platform to experience nature in an enhanced dimension.
Good evening to all participants. It's a great pleasure for me to introduce Dr. S. Jayakumar on today's international webinar on behalf of BCF. So Dr. Jayakumar is currently serving as an assistant professor in Department of Zoology and Wildlife Biology at A.V.C. College, India. And he has completed doctorate and specialized in organochlorine pesticide population and reproductive ecology of fish-eating birds in Tamil Nadu from Salimali Center for Ornithology and Natural History, SACO. And he has completed MPhil in Zoology and double masters in Environmental Science and Zoology. And adding on towards his publication, international publication in peer-reviewed journals and 6 for the national level. And he has co-authored for several books and book chapters. And Dr. Jayakumar has won Best Teacher Award from Directorate of Education Tamil Nadu during 2006 and 7. It's great to have you, sir, today. And he also won several awards for his international publications for his conferences. So Dr. Jayakumar is currently serving as a reviewer for Journal of Ethnopharmacology, Elsevier, and Journal of Traditional Medicine, Bentham Sciences, and International Journal of Veterinary Taxa, JOT, for Wildlife Society, and Zoo's print magazine. On behalf of BCF India, I welcome Dr. Jayakumar for his international webinar on Impact of Pesticides on fish-eating birds in Tamil Nadu. Over to you, Dr. Jayakumar. Thank you all for all participants.
Madam have very nicely introduced myself. Yes, I'm a village guy actually, born and brought up in a small village that even not known by others, which is called Mudikandanallur, Manalmedu. So I actually studied my schooling and all, there in government higher secondary school in Manalmedu. And then for me, like I thought, where do I do my higher education? So in that point in time, A.V.C. college, it was a really a good one where I could join there and then completed my masters and all there in A.V.C. college. This is where our department lies. We do teach both zoology and wildlife biology. And from there, I do not know how do I pursue my research, because I wanted to do some research basically. But I am actually lack in all the resources that where I can get my higher education or research and all, because being a village guy, I was so remote in that couldn't able to communicate with the entire world. So at that point in time, I came to know there is a research institution called the SACON through my professors, and my professor insisted upon me to go there and attend the interview. So there I have attended interview and then joined as a technical assistant in the project, and then later I promoted as a JRF, SRF, and in a project funded by Ministry of Environment. So I got my PhD there actually. So this is what a kind of small introduction about myself. Okay, let me talk about the topic like impact of pesticides on fish-eating birds in Tamil Nadu. So what is pesticides? Most of you know that it is a kind of substance that are meant to control plant enemies. Who can be the plant enemies? What are all the targeted species that we consider as a plant enemies? You may be say like weeds, the unwanted weed which is actually growing along with the crop can be considered as a enemy. And then obviously rodents, you can say this is one of the enemy. And the insects, next to other population, insect are a huge one. When you look at the population of insect, it is it is really hard to control them actually. So yes, we do have a couple of species of insect which is damaging the crop and then damaging the yield of a crop, and then even couple of microorganisms like bacteria, fungus. So those are all act as enemies against the plant. To control that, people have found out a chemical called pesticides that actually we would get to know the major area of pesticide use in India. I think I'm audible to everybody. Like, yes, we have, two major sector where we are using pesticides. One is agriculture, which constitute around the 69 percent of usage, and then remaining one is civic health purpose, like public health purpose. In order to clean, malaria causing vectors and all, we do use chemical to control the malaria causing vectors like mosquitoes. Yes, the story of usage of pesticides in India began in the year of 1948 through DDT control malaria that everybody know. And the next, for agriculture purpose, like we started using BHC, benzene hexachloride. That is a major area of pesticide use actually. So no, India has a history of using DDT in order to control malarial causing vectors called the mosquitoes in the year of 1948, followed by use of benzene hexachloride to control locusts. When I talk about locusts, you people more aware about it actually. Yes, this is what these are the major sector in India started using pesticides actually. And then again, if you divide the pesticide based on the target, it can be classified as a fungicide. It can kill fungus. Insecticide, it can try to concentrate, kill insects. Herbicide, yes, unwanted weeds. Nematidicide, rodenticide. No, it goes, a number of times according to the target of the pest. When you talk about the percentage use of such chemicals in India, obviously insecticide contributed higher level, particularly the 76% of the chemical that what we use or the farmers use are insecticide to control insect because they are the problematic insect to damage our crops, followed by fungicide and then herbicide. So these are all the percentage by and large we use. And then yes, there are a number of chemicals. When you ask a farmers, he may say karate, kung fu, insect killer. There are kind of pesticide names actually. It is so branded in fancy names so that it gets attracted the farmers actually. But for me, I wanted to classify the chemicals according to the chemical nature. They are actually classified into four actually. One is organophosphate, organochlorine, carbamate pesticides, and the pyrethroids chemicals. So here, for me, concentrating on all the chemicals would be a bit difficult. So that is the reason I thought, that of working with a legendary chemical. What is called a legendary? The legendary chemical in the sense that they are so persistent in the environment. So when you apply today, even it may be exist for more than 25 years actually. So we need to work on such persistent chemical because the effect would also be persistent to ecosystem. So by considering that, I thought like I could work with the organochlorine insecticide. So though there are kinds of pesticides we have been using, but I worked only on organochlorine. The reason behind they are so persistent into the environment. Yes. How chemicals are entering into ecosystem? That is one question most of the participants would ask. Is it a food chain accumulation? When you ask, my question is, my answer is that no, yes. So whenever we use chemical pesticide by using any kind of sprayers, like you can use box sprayer or backloaded sprayers. Whenever you apply the chemicals, that would actually into agriculture field adjoining to the bird sanctuary, where the birds are breeding in more numbers actually. The pesticide run into agriculture field, run off from agriculture field actually get into the sanctuaries where the fishes are depending on insect as their major food actually, so that the fishes get accumulated into such chemicals. When fishes are getting affected, like when fishes are picking those pesticide residues from water, you may imagine like even the birds who is feeding on fishes can also get accumulated through food chain accumulation. Either way you can do, either by the way of documenting the chemical level in fishes, you can able to assess the contamination level of any bird sanctuary. Other words, you can able to do research on birds and then trying to assess the contamination of heavy metal or any pesticide content in the bird sanctuary. So this is how the pesticides enter to the field actually. And then you may ask, sir, why you have selected only fishing team birds? There are a number of birds there in India. Yes, before that we could get to know the effects, the major effects of pesticides on birds. Yes, in case we apply pesticides, sometimes it can kill n number of birds. That would say like mass care of birds, mass mortality. We may get to see a death of birds almost like in 1,000, 2,000. When it happens, that may be considered as a mass care. You cannot able to control that actually. And then the pesticide may cause reproductive failures. It may not kill the bird directly, whereas the small concentration can cause the reproductive failure among very many birds in India as well as elsewhere. And then it also tend to cause eggshell thinning that you know very well. No, there is a one chemical called the carbonic anhydrase, which is an enzyme which is responsible for production of calcium carbonate in bird's body. What such chemicals will do, they go and bind with the enzyme, it will suppress the activity of that enzyme. When the activity of that enzyme suppressed by the pesticides, the bird may not be lay proper eggs, means the eggshell would be get thinning due to a pesticide called DDE. So due to pesticides, even we will be getting eggshell thinning. Sometimes, yes, egg will be doing good, but we would observe the chick mortality. So whatever the picture I have shown would definitely give you the evidence of effect of pesticides. So we could also see the chick mortality actually. We may know that egg is good and it may be hatched, but unfortunately the egg may not be hatched due to pesticides effect. And chick edema. Edema means couple of couple of organs of a chick actually bloomed up or grown up in abnormal way actually. The abnormal growth, we may say that is an edema. It is a kind of disease which is actually caused by a pesticides. And then we could also have a hatching failures by pesticides, and then we may have a embryo mortality. Entire embryo may die due to the pesticides. There is a chance of egg breakage. My dear friends, you may know that the birds called white-packed vulture or white-rumped vulture used to lay only one egg per year or two. So in case the pesticide causes the effect to even that one or two egg, you imagine the fate of their population in future. So this is what happening. And then yes, there might be changes in physiological and behavioral activity as well. What is that physiology and behavior? Say for example, whenever any defense, very often it happened to me, I used to defend myself by the way of n number of enzymes. There, bird may not have such opportunity due to chemicals, because these chemicals will go and bind with such enzyme and then suppress the activity. When that happens, bird may not be able to exhibit the regular behavior that they do generally. And then the drooping head, you could see the picture there. Hope everybody can see my slide. This is a white-backed vulture having drooping head. Drooping head is a abnormal behavior that we could watch it in the wild when the birds get exposed to pesticides actually. So yes, pesticides causing so many problems. I could able to list only few problems that I have noticed in my field, in my research. So these are all the problems that pesticide causes. And then yes, the researcher should explore what is that and how chemicals causing impact on birds. Every researcher should watch that actually. So in that manner, how many species get been explored by researchers? We must know this actually. Here I wanted to pay important to Dr. S. Muralidharan and Dr. Dhananjayan and their team from SACON actually. So when I was doing my MPhil, I did not aware that there is so many research going on inside SACON on impact of pesticides on birds. They did research on chemicals, influence on vulture population. They also did research on kites and then grey-headed Himalayan eagles, and they also done research on crows. They have done some research on water birds, particularly like pin-tailed duck, which used to migrate from North American Mongolian countries. And then yes, they also researched on a small passerine bird called house sparrow. Like they also documented that the effect of chemicals on house sparrow, and they also worked with a dancing, excellent dancer, the Saras crane, that used to dance like anything with a pair. Whenever you go into a field of Uttarakhand or some central Indian region, you may be amazed to see their dance actually. Yeah, tall bird it is actually used to dance like anything. Even such birds are also vastly affected by chemicals. And then again our national bird, peafowl, also getting vastly affected by the chemicals. So these are all the bird species which were really studied very well or established very well by Dr. S. Muralidharan and then Dhananjayan and their team. We should appreciate their contribution towards just producing the information about the impact of chemicals on such birds. And then yes, after completing my MPhil, I thought of doing research, so I just landed SACON. When I landed SACON, I was fortunate to be join with the team with the Dr. Muralidharan and the Dhananjayan sir. So when I asked, sir, I just wanted to do some research, they said like time to go and collect literature review. Yes. So myself and my boss wanted to collect literature review, in which we took a truck and then wanted to travel around the world. When our truck traveled in the world, like particularly when our truck arrived there in North American countries, we got truck full of information. That means the North American scientists are so good that they have really, tested or assessed the impact of chemicals on birds. That is the reason we got a truck full of information from North America. And then, we wanted to come around the European country by using our truck. Again, our truck got some amount of, literatures. Those people also done good research. Unfortunately, when our truck landed Asia, we did not get much information on effect of chemicals on raptors, water birds, and fish eating birds. And then yes, of course, when our truck arrived in India, we got some information about the pesticide effect, but not on fish eating birds, on some other species. And then I came to know that there is only one study done by Dr. Muralidharan, who his research actually on eggs, particularly on eggs of fish eating birds in Kledev National Park, which is there in Bharatpur, Rajasthan. So to me, it is a surprise that I have got only one study in India which is pertaining to fish-eating birds, effect of chemicals on fish-eating birds in Tamil Nadu. That point in time, when we are returning back to Sakon, our truck was there in around Chennai or other places. My boss, Dr. Muralidharan, asked me, "Where are you from?" I said, "I am basically from Tamil Nadu." What he did, that he just pushed me away from the truck and asking me, trying to set the objective, because from your own state, we don't have any information on the effect of pesticides. So then I realized, okay, I came to do research through SACON. My boss has given some assignment. By thinking that, I just set this objective. I do not know whether it will fit or not, but I just simply set this objective. First, I wanted to conduct the surveys and locate the hernaries of Tamil Nadu. Initially, we wanted to do research on fish-eating birds, but during my field visit, I came to know fish-eating birds and non-fish-eating birds together, they are breeding. So when they breed together, they are called colonial nesting birds. So instead of looking at only fish-eating birds, later I modified my objectives, looking into even the colonial nesting birds, the birds which used to nest together for protection. They are called colonial nesting birds. That is what my first objective I have selected. And the next, we need to monitor the breeding ecology, because then only we can get to know whether the eggshell thinning is there or breeding impairment is there or not. That need to be noticed when you closely watch them, their breeding outcome actually. So I did monitoring a breeding ecology and the nesting success and mortality of birds. How many birds are actually getting died due to pesticide or even any other reason, that I have assessed in select location, because assessing all the information from all the bird sanctuaries in Tamil Nadu is not possible actually. So within a shorter period of time, so that we have assessed only selected regions, and then we also document what sort of documenting the residue levels of persistent chemical in fishes. Why do we have to document chemicals levels in the fishes? Because I'm concentrating on fish-eating birds by and large. So through their food, they can able to get they can able to pick up the pesticide. That is the reason I have done pesticide analysis in fishes, eggs of birds, and the tissue of dead birds. We never ever kill any birds actually. Whenever we found any dead birds across the sanctuaries, we used to collect them actually for pesticide analysis. And then we also want to find out the relationship between eggshell thinning and population decline. I said no, eggshell thinning is one of the problems. Whether it is really impact the entire population of birds, for which we need to have some assessment, for which these are all the objective that I have fixed. Again, we need to select the study area. Though I have visited the entire bird sanctuary in Tamil Nadu, I have selected only 3 bird sanctuary as my intensive study site. One is Vedanangal Bird Sanctuary, which is there closer to Chennai actually. Another one is Vatangudi Bird Sanctuary, which is there near Madurai. And then Kunnangulam Bird Sanctuary, which is dead Tamil Nadu. Why you have selected only these sanctuaries? Why I have selected only these sanctuary actually? So you can ask actually. You can see the linear pattern of selection. Statistically, when you're trying to select the linear pattern of selection, that would be support as actually. And then what are the other reasons behind selecting these sanctuary as an intensive study site? Yes, it may be easy to go and study, and then total area is good, and these are the lat lawn that we have noted. And most interestingly, more number of fish-eating birds and other birds are breeding in these sanctuary when compared to the rest of the sanctuaries in Tamil Nadu. Vedanangal, we could get to see 18 fish-eating birds, 9 other birds breeding, and then it is there very well abundant. 21 fish-eating birds, 14 other water birds in Kunnangulam, 15 fish-eating birds, and then 7 other water birds in Vatangudi Bird Sanctuary. Breeding seasons varies across. By and large, it starts from November and then it ends with sometime February or March. Even it can be extended up to April when there is a failure of monsoons. These are all the breeding birds. I can give an in short name, actually, little cormorant, Indian cormorant. If some of you wanted to know in detail, I could discuss it little later. And then we also assess the nesting trees for such bird sanctuary, how the nesting trees are supporting the birds for breeding. So yes, fortunately, Vedanangal had a good number of species, which includes of Acacia nilotica and Barringtonia catangula, whereas even Kundangulam do have Acacia nilotica, Asgartica indica, Prosophis juliflora. Though it is, we may see that it is an exotic breed, but Prosophis juliflora is excellent substrate for the birds to breed, particularly in all around all the habitats. Vetangudi do support couple of plant species for the birds to breed actually. Again, the major aspect of selecting these study area is just based on the agricultural activity. When I look at the agricultural activity among all the three bird sanctuaries, it was so good. The major agriculture, the plant that they grow near the sanctuaries are paddy, sugarcane, paddy, plantains, and paddy, black ram, according to the sanctuaries. So based on the Cali, 2002, the criteria, I have selected these three sites as a potential site, because when I want to look at the breeding impact, I need to look at the area where the birds are breeding. So that is the reason I have selected the top 3 bird sanctuaries in Tamil Nadu. Vedanangal was the oldest bird sanctuary of India even. So I have selected that. Kundangulam, yes, it is located dead south. It may be simply called the painted stock village. Kundangulam can be considered as a painted stock village because there are around 7,000 painted stock used to be there in the area. And then Vetangudi, which is so close to Madurai, the reason began selecting that area, even there some agricultural activity there in Vetangudi as well. So based on the agricultural operations and most number of birds breeding abundance, I have selected those 3 sites as my intensive site. Yes, I have assessed the information based on a couple of methods. For population estimation, that direct count method is the apt method that I have followed. So many formalities are there, like binocular used, care was taken care, population estimation done both the breeding season as well as non-breeding season. These are all something about assessing the population of fish eating birds. And then next, breeding biology. We need to monitor the breeding biology. So the monitoring effort involved the following major step called first I determined the area by nesting panorama, the sketch map of a nesting site or nest. That is nothing but nesting panorama. I created the nesting panorama and followed the focal nest sampling. The area visited frequently to assess the nesting activities during all the season, both nesting as well as non-nesting seasons, right? So these are all the methods that I have adopted for assessing ecological information, ecological data. And then yes, on toxicology, to assess the effect of chemicals, we need to collect the sample from the bird sanctuaries. Yes, so I have collected the dead birds from the sanctuaries and did postmortem there in the field sometimes, and I did postmortem there sometime in the laboratory of Salim Ali Center, Coimbatore. So I have collected both the dead birds as well as the fish sample. So the tissue sample, whether it is a bird tissue or fish tissues, ground very well with the sulfate, sodium sulfate. It is a salt by which can able to remove the moisture from the sample, and then did for Soxhlet apparatus, Soxhlet extraction for extracting the chemicals for 6 hours, extracting the chemicals from the mussel by using Soxhlet extraction, and the later column cleaned. We used 2 fraction, 1 fraction for polychlorinated biphenyls chemical. It is yet another organochlorine chemical which also pose serious threat to birds, and then fraction 2, F2 for pesticides. So the condensed sample analyzed by using GC, Hewlett Packard 5890 series 2 equipped with the ECD. This is the equipment which is available very well there in Salim Ali Center. And then for eggs, I cut opening the egg sample and took the content, measured the eggshell thinning for thinning activity, and then comparison. And then those content were column cleaned and then condensed again, and then the sample were again analyzed by using gas chromatography. So I'm now just trying to cut short the techniques, because in case if you want to discuss in detail, we will discuss in the discussions. Right. Coming to the results, yes. So I wanted to get to know the effect of pesticide or the levels of pesticide in fishes, dead birds, eggs. We have also assessed the level of pesticides in water, level of pesticides in sediment, level of pesticides in some other food as well, but that I'm not presenting right now. So because when I talk about fish eating bird, I restrict with a couple of components called fishes is their major food actually. So I want to project the effect of chemicals on fishes, later into birds, and then finally in egg content as well. So these 3 components only we will be discussing in the slides. Yes. How many species of fishes I have collected from these three sanctuaries? I have collected almost 9 species of fishes from all the bird sanctuaries, all the 3 bird sanctuaries. From Vedandangal, totally I have collected 124 fish individual, which belongs to 9 species, maybe Labia rohita, Anaphas textudinus, Silginus migala. You could read all the fish species, and then proper gearing net, net followed to catch the fishes. We also consider like the net can catch equal size of fishes, or the net can catch even small size to moderate to big size of fishes actually, because birds tend to consume both small fishes as well as large fishes. By the way of assessing only large fishes, we should not say that this pesticide is influencing the birds. We need to get entire size fishes, for which we adopted different gears, different method to catch the fishes actually. From Kundangulam Bird Sanctuary, around 74 fishes collected, and then from Vatangudi Bird Sanctuary, 104 fishes collected. Here I have given the length and the weight of the fishes along with their feeding habitats. Why feeding habitat is important? That is important because, like, when the animals are there in the top tropical level, they tend to accumulate more chemicals. For that reason, one need to assess the feeding habit of fishes as well. Levels, organochlorine pesticides in terms of ppb in fishes of Vedandangal. So I'm analyzing set of data, Vedandangal separately, Vatangudi separately, and Kundangulam separately, because the fish species found, those sites are not uniform. That is the reason I analyzed separately. Later, whatever the fish species found in all these 3 locations, I took it for comparative analysis among the locations. So here, when we talk about the fishes of Vedandangal, Anaphas textudinus had a more concentration of total HCH. What is called HCH? When you say total HCH, which include your 4 chemicals like alpha HCH, beta HCH, delta HCH, gamma HCH. It has 4 isomers. Put together, I actually sum up all the isomers and then declare, trying to represent as a total HCH. Same way, total endosulfan. Like, endosulfan A, alpha, endosulfan B, beta, and then endosulfan sulfate. Put together, I am presenting as a total endosulfan. Because whenever I present the individual chemical, it won't be look good, because I have done around 16 chemicals. So projecting results in relation with all 16 individual chemical won't be ideal, and then it may bore you as well. That is the reason I sum up everything and then trying to give you total DDT. Here means like DDD, DDE, DDT. So it is all actually kinds of metabolites put together. I'm trying to present heptachlorepexide. HE means heptachlorepexide. It is another chemical, and then dialed. So when you look at the result, actually, fish Anaphas textudinus had higher levels of total HCS, total DDT, and the dialed. So you could see the fish species. My dear friends, I wanted to quote you one more information here. The Anaphas textudinus, when you go and see search IUCN website, you may be shocked, because this fish is data deficient fish, data deficient DD. They have rated in IUCN, which means we don't have a proper information. We don't have a proper any information about this species, but this species get accumulated into more organochlorine chemicals. That is what my study reveals. So they are the most accumulated one when compared with the Oreochromis mozambicus and the Mistus vitatus with the rest of the chemicals. And yes, this graph would explain us the effect of chemicals or the levels of chemical in the fishes, which is collected from Kundangulam Bird Sanctuary from Tirunelveli. Again, there the fish Mistus vitatus. This is the most tastier one actually when you eat actually. And then this very good fish species for egrets, they can able to easily catch, because the big size of egret and then the fish size is almost the same. So that most of the egrets would love to eat these fishes, unfortunately contaminated with all these chemicals, followed by Labia rohita, which is again a herbicide. This one like herbivorous fishes. And then this graph showing the information about the pesticide levels in Vatangudi Bird Sanctuary, which is located in Madurai. There, Oreochromis mozambicus, the fish actually is the most abundant fish of freshwater. Even it now developed the adaptability to live even in the brackish water actually. So this fish had more concentration of total HCH and endosulfan, followed by Labia rohita. So instead, I just wanted to summarize the levels of fishes, levels of chemicals in the fishes. So when you take total organochlorine chemicals, total HCH recorded in higher level, followed by total DDT, endosulfan, and so on. Which means this, the summarization giving us total HCH was the most frequently found chemicals in the tissue of fishes than DDT. DDT, so it is even banned chemical that is being noted in the tissue of fishes. It has been banned. DDT has been banned in India, but we still getting the residues from the fishes. And then when you look at the species wise, Anaphas testudinus, the data deficient fish accumulated more amount of chemicals than Oreocorampus mozambiques and then Channa functatus. And when you compare the locations, Vadanangal bird sanctuary, so-called birds of paradise, accumulated more chemicals. The fishes there are actually not safe for birds to consume because that has more chemicals in it than Kundangulam and Vattangudi. Also analyzed the season-wise variation of chemicals in the fishes between breeding and non-breeding season. There is no much difference I have observed from breeding and non-breeding season, which is not statistically supported. And then yes, isomer I said in the beginning, beta, alpha, gamma, delta. Among all the isomers, beta contributed more when compared with others, whereas among the metabolites, DDE contributed much than the DDT. My dear friends, here you must note one. DDE is the chemical which is not actually manufactured by anybody. The manufacturers, pesticide manufacturers, only manufacture DDT. And then, like, when you take cyclodine insecticide, which are all the, mass classification of pesticide that you don't have to worry right now. So, in which, the endosulfan, the other chemical, which is nothing but organochlorine chemicals, contributed more. And then I compared the concentration of, pesticide in the fishes in India with studies elsewhere in India as well as other countries. And then most interestingly, DDT recorded in higher than level prescribed for fish products. Like, FAO and WHO given some, prescription, like, the concentration should be, the DDT concentration should be 2.5 ppb. In case if it exceed, it would cause some problem to the fishes. That is what WHO recommended. But now the fish species which we have analyzed, all the fish species are exceeding the limits which is actually given by the WHO. The level is 2.5 nanogram per gram. And then, like, there is another shocking news that, Novel in the year of 1987 have reported the guideline values of, 200 ppb of total DDT in fishes for the protection of colonial fish eating birds. What it indicates, when the concentration is going to exceed in the fishes like more than 200 nanogram per gram, that means when the bird is going to eat the contaminated fishes or the fishes which is having this concentration may be harmful to the birds actually. So it is harmful to the fish eating birds. This is what the guidelines the WHO have given. So it is actually again adding up our curiosity to get to know how birds are responding against the pesticide, for which we have wanted to collect the fish eating bird sample actually. Before that, whatever the fish, the levels that we have reported in the fishes have also been published internationally. The levels of organochlorine pesticides, residue in freshwater fishes of 3 bird sanctuary in Tamil Nadu. This is the first study reporting the levels of chemical from the protected area in India. This is the first study of reporting the chemicals in the fishes of protected area. So this is the pride that we have actually. And the next, yes, when the fishes are suffered with the chemicals, and then what about the fish eating birds, which is depending on fishes as their major food, for which we wanted to assess the dead birds, because we have any right, we don't have any right of killing the birds actually. So we have collected only the dead birds as an opportunistic sampling method. Opportunities, whenever you get dead birds, you can collect, for which we need to thank the forest department. They have given permission. Even after trying so many efforts, we got a permission actually. So around 14 species of this I have fish eating birds I have collected. We should not say it is a fish eating birds, we can say colonially nesting birds, because even when you look at Asian openbill, they are not a fish eating bird, they tend to eat mollusk more in numbers. So that is the reason I said, instead looking at only the fish eating bird, later we modified our objective of looking at the entire colonial nesting birds, the birds which used to breed together in an area. They are called the colonial nesting birds. So these are all the number. Unfortunately, we did not have more dead, we did not received more dead birds actually. So from Vattangudi, we received 10 dead birds. From Kundangulam Bird Sanctuary, we received 63. And then, from Vedanangal 10, Kundangulam 63, and then Vattangudi 3 individuals. So based on, by using all these individuals, we have gone for analyzing the levels. And then I'm now trying to give this graph, which is only meant to with the herons and egrets. So among herons, like Siconiforms, Siconidae when you say, among the herons, cattle egrets suffered much with all the chemicals. followed by Indian pond heron. My dear friends, we think that Indian pond heron is, we think that the Indian pond heron is the most abundant one. Yes, it is most abundant one in the past, not right now. Because when you go and see, it is called the paddy bird. The paddies you could see without this bird actually. Though the name we need to change actually. So this bird is getting mostly affected by the chemicals called HCH and DDT endosulfans, followed by night heron. Night heron, they used to separate their niche along with these among with these herons actually. They used to forage only night hours. Even their behavior is now get changed due to a number of changes in the agricultural activities. They do get mostly affected by chemicals like HCH. And then other graph is actually talking about the effect of pesticides on storks, ibises, and spoonbills. Here, among all the other birds, like long-necked birds, we can say Eurasian spoonbill had a higher concentration of total HCH, total DDT when compared with the rest of the bird species. And then this graph actually giving us the information about the effect of pesticides on pelicans and cormorants. Unfortunately, little cormorants are not doing good. Little cormorants used to be huge in number in all the bird sanctuary. Whenever we visit Tamil Nadu, we used to sight plenty of little cormorants, and the little cormorant population vanished due to Sri Lankan war. That is a hypothesis that is going on because there's a Sri Lankan war there actually that actually disturbed this bird. And I would say here authentically by using my own data, more than the Sri Lankan war, the pesticide caused a severe problem to little cormorants as well. There's a huge number of birds we couldn't be able to see just because of the effect of chemicals. And then again, the Indian cormorants, Indian sac, we say in other words, which is also not doing good against the chemicals. Then the spot-billed pelicans. And then again, when you look at the contamination level, like I am trying to give the summarized version and the discussion for you to easily understand. Yes, as I mentioned earlier, among all the chemicals that I have analyzed, total HCS had the highest level than the total DDT. Whereas when you look at the species-wise comparison, obviously cattle egret, the bird which is depending on insect as their major food, they used to forage around the agricultural land actually. They used to go with cattle. It is nice symbiotic relationship that we get actually along with the cattle. That bird is not actually doing good against the chemical. They actually accumulated more components than grey heron and Indian pond heron. My dear friends, look at the species, cattle egret, grey heron, Indian pond heron. Those are all the species completely depending on agriculture as their major foraging ground. So that is a clear-cut indication that the chemical which is being used in the agriculture are directly affecting the birds like cattle egret, grey heron, and Indian pond heron. When you compare the location-wise differences, Vedanangal had higher levels of pesticide than Kundangulam and Vettangudi. You look at isomers, again, beta isomers is more. And then when you look at the metabolites, DDE concentration higher when compared with the rest. And then when you look at cyclodine, total endosulfan actually causing serious problem. Then heptachlor and dieldrin. Yes, for the discussion point of view, I wanted to give you that actually. The residual level of HCH and DDT, dieldrin, and the rest of the chemicals detected in the birds said it do not indicate any poisoning. As of now, these birds actually not actually doing, it is good actually, they are doing good. The chemicals are not causing serious problem, but when you put together because I am discussing individually, when you add all the chemicals and then look at, definitely it would cause serious problem. And then the levels which I have detected in Tamil Nadu, I have compared with the rest of the countries like in Greece, India, and then like Italy, Japan, London, Netherlands, South Korea, and Thailand. And yes, when bird will die, sir? You may ask, when bird can die due to chemicals? Yes, when the DDT residue level in all the birds are lower than the critical concentration, which is represented as greater than 30 ppm actually. When the liver consists of more than 30 ppm of DDT, the bird will die. But it is so fortunate to us actually, none of the bird which we have analyzed crossed the limit, whereas the fishes crossed the limit. That we need to note in point. Yes. So when we look at the chemical compositions, like again, the beta HCH, wherever I represent, beta HCH contributed more. Why? What is the reason? We say like beta HCH detected in more level, more tissues. It is more in tissues, more in fishes. Why? What could be the reason behind? Because the alpha HCH and the gamma HCH may convert in the form of beta HCH. That is the reason we are detecting more levels of beta HCH in all the tissues. And moreover, the beta HCH, the chemical, which is more persistent than rest of the isomers. That is the reason we are detecting more levels of such chemicals in both fish samples as well as bird samples. And then again, among the isomers, among the metabolites, like when you say DDT, DDE, and DDD, among the metabolites, like DDE had higher concentration. The reason began, as I said earlier, they are persistent in nature. None of the manufacturers are using DDE, whereas the DDE actually coming from DDT, which is a transformation effect actually. So DDT will convert as a DDE in the environment. That is a most notorious problem. And again, when you look at the comparison, like the effect of DDT will not be much, whereas when you look at the effect of DDE is more. So we need to really concern about the effect of DDE on birds. So when we assessed around 50 to 70% of the contribution is done by DDE, that is the most shocking news that we got when we analyzed the samples. And then among the cyclodine insecticide, the fancy chemical called the endosulfan had a higher level in all the tissues of birds as well as fishes, followed by heptachlorhexide and dialtrin. So we also published this actually in the Journal of Archives of Environmental Contaminant and Toxicology, which is a so prestigious journal, so tough enough to publish that our paper in the journal. And again, I could read organochlorine pesticide residues among colony nesting birds in Tamil Nadu, a maiden assessment from their breeding ground. When I said no fish-eating fishes, that was the first study I mentioned. Later, when I'm trying to say about the documentation of the fishes, the birds, again, this is the first study from India, from the protected areas. It is a maiden assessment from their breeding grounds, actually. And then went on analyzing the egg samples. We collected unhatched egg from the nest, and then sometime which is actually fallen from the nest used to float on the water. I used to collect the eggs and then watch it, and then I trying to identify by using the egg guides. There are a number of egg guides available in Salim Ali Center Library. Based on that, I have analyzed I have identified the egg samples and then done the chemical analysis. So starting Asian open bill stock, black crown nightingale on Eurasian species, Eurasian spoonbill. Even I have collected eggs of spot-billed pelican, which is actually a near-threatened birds, and then you can say painted shark is a near-threatened bird, and black-headed ibis, which is a near-threatened bird. All unhatched eggs, which is actually floating on the water that we have collected, identified, and then used for pesticide residues. So again, from Vedanangal, we got 34 egg samples. From Kundangulam, we received 52, and then from Vattangudi 11. Overall, around 97 eggs that we have used. And this, the table, which is giving us the information about the effect of pesticides on fish egg actually. So among all the bird species, the species intermediate egret, we have 4 species of egrets which used to commonly visit Tamil Nadu. So among all the bird species, the bird which is intermediate egret had higher levels of chemicals, followed by little egrets, and then Eurasian spoonbill had the least level of chemicals. What it indicate again, the birds which is depending on agricultural habitat as their major foraging ground are picking up the chemicals, and their egg, when you look at the contamination level, is more in their eggs. Yes. And then I wanted to analyze, I wanted to look at whether these residues can cause any eggshell thinning. So I did a correlation analysis. The correlation reveals that there is a negative trend, which means where there is a DDE level, which shows the decrease of eggshell thinning. But we don't have any data which is supporting statistically. The reason behind, we have got only lesser sample size. Probably we need to increase the more sample size, get to know statistically, yes, there is an effect of DDE on eggshell thinning. But still we could say there is an effect of eggshell thinning problem by the pesticide called DDE and dialtrin. And then the most interesting part that I wanted to discuss with you. How do we relate to the effect of pesticides with the breeding success? For which we need to adopt a single egg sampling technique, which is so tougher to convince the forest department to get the permission to take one live egg from the nest, for which we got minister permissions. Due to minister, we got permission from PCCF, and then we have collected one egg sampling. What is called the one egg sampling? When the bird lay first egg, we used to monitor, and then we go and collect the egg during the night hours. Myself, swim into the bird sanctuary with headlight, and then collected the one egg, I used to tag the nest actually and then come into the ground. So after collecting the one egg, one may wonder, sir, why you are collecting live egg actually? Bird will again see the nest and they may thought like they did not lay the egg. They will again lay the egg. They will try to complete the clutch. We will not disturb the bird in such so that bird can continue their clutching. They can complete their clutch size. After that, the collected first egg we have analyzed for pesticide chemical. This is how we can able to compare the effect of pesticides on breeding success. In which, most interestingly, I wanted to tell you, my dear friends, black-headed ibis, the near threatened bird. When the egg of black-headed ibis, the near threatened bird had around 72.74 ppb of DDE, one chick found dead in the nest, which means the remaining, the leftover eggs which is there in the nest, we used to monitor continuously. When we look at that, one chick found dead. Yes, we can relate that may be due to the effect of chemicals and subsequent foraging of contaminated food materials. Likewise, even there is another bird called the painted stork. When the level is actually 228 ppb, again one chick found dead on the nest. What would be the reason behind? We have actually collected the chicks from the nest, and then when we analyzed that, it has a chemical contamination, and maybe the bird died due to chemical contaminations. So this is what happening. So yes, this is a very good authentication that again the chemicals are causing serious problem to birds. For you, I have summarized this again, like among all the chemicals, total HCS contributed much. When you look at all the species, intermediate degrade again suffered more. Among the locations, Vedanangal had higher levels of contamination in the eggs, which I have sampled from Vedanangal, and then like isomers, metabolites, cyclodins, which we have also published in the book by Oxford University, Threatened Birds of India, their conservation and requirements, edited by Dr. Asad Rahmani. So our chapter actually termed as status of pesticide contamination in birds in India with special reference to threatened species. I have only summarized only the threatened species like painted stork, spot-billed pelican, and then black-headed ibis. The rest of the bird species yet to be published. I'm actually in the process of publishing the same. Most probably we would get the same publication within this year, so that we can have a more authentic information. You may ask, sir, you have been talking about the effect of chemicals. What would be the solution, sir? How do we stop it? How do we escape? How do we conserve these birds from the effect of chemicals? Yes, we can do integrated pest management, IBM we say. So we use that actually, so that the usage of chemical could be reduced, and then we do try organic farming. That is what our forefather Nammalvar has given the way. Actually, we need to go for organic farming. We should not rely on the green revolution based modern agriculture. Yes, we need to protect our we need to take care of our production in terms of to meet the population, but that should not the cost of ecosystem. That should not at the cost of species. That should not at the cost of fish eating birds. So we need to do organic farmings, and we should also be in a position to use chemicals rationally. That is what called rational use of pesticide, RUP. Most of the developed country use that patterns. They used to go for rational use of pesticides, RUP they follow. But in India, we never ever follow. Even it is wonder that when you go and ask to a farmer what chemical you are going to apply, a farmer may say like the green bottle, the yellow bottle, the white bottle. My fellow farmers are unaware of even the chemical name. That is the case. How we are going to educate the farmers? That is what the most important issue with us. So yes, we need to create awareness among the farmers about the effect of chemicals on pesticides. I mean, on all kind of chemicals, and we need to use the biopesticide. We have to use biopesticides, which is actually not causing any problem to any kind of organisms in the field. We need to construct the earthen dikes all along the bird sanctuaries. So among all the three sites, except Vettangudi Bird Sanctuary, both Kundangulam and Vedanangal did not have a proper earthen dikes all around the sanctuaries. One area it is open up to the agriculture land. When there is a rainfall from the agriculture land, there is a water running into a bird sanctuary. It is not the water running in, it is the residue which is running in from the agricultural land actually. So what we need to do, we can construct the earthen dike so that the agricultural runoff into the bird sanctuary could be stopped by and large. And then yes, we need to actually fastering our research. We have done some research, but we need to faster our research. This won't be sufficient. We have concentrated only on fish eating bird. Even there are rest of the other birds are suffering. We need to work on that aspect as well. So we need to fastering the research aspect. And then yes, of course, where the birds are breeding, there we need to put the restriction that the farmers should not use application of chemicals within 50-kilometer radius. So when they stop, usage of chemical, at least the winged visitor can escape from the chemicals. Yes, there are a number of natural predators like spiders, like dragonflies are predate the pest. We should promote. We should actually ask the farmer to look upon the natural enemies, and then we should stop usage of chemicals, and then we should go for we should always rely on our forefather Nammalvar words whenever we do agricultural activities. So when we do that, definitely we could able to come out from the issues. Yes, my dear friends, in the beginning, you may know that when our truck arrived in North American country, we got truck full of information. European country, we had some information. Asia, we did not have in India. Yes, we have some information, but on other species, not on fish-eating birds. So in my previous slide, I decided that from Tamil Nadu, there is no study. That point in time, my boss asked me to do the research. Yes, I thankful to my boss. He asked me to do the research. I have done it. And yes, now from Tamil Nadu, there is some data we have published. Yes, we can fill the gap. We have filled the gap. We are going to fill the gap even by using our future publications as well. So who have helped to complete this task? Yes, this task is not easy to complete by myself alone. There are a couple of team who has helped me to generate data from Tamil Nadu. So I am from Tamil Nadu. I have generated data for my own state. Like you need to generate data on the effect of chemicals on your own state. In case if you not, please contact us. Please contact our team. Please contact my boss. They can able to help you to assess the effect of chemicals on even other bird species as well. Why do we do other species, even fish-eating birds in other state as well? So that when we compare the effect of chemicals on fish-eating birds rest of the state, we could able to come to a conclusion that yes, if you want to conserve this bird, you can adopt this. Yes, for which many teams have helped. I just want to thank my research supervisor, Dr. Muralidharan sir, who is a principal scientist there in Salim Ali Center, who actually who trusted me to do this research. And I also want Dr. Dhananjay, my senior, who helped me in terms of, for other aspects. We wanted to thank Ministry of Environment and Climate Change. They have, given fund to do such a research. Without their fund, definitely it may not be achieved. So here we take in turns, thank agree to the MOEF for supporting us financially, and then of course Forest Department. Without them, this work wouldn't be possible. They permitted us to go collect fish samples, though it is a protected area. They asked us to collect the egg sample and then dead birds. So we need to thank them as well, the Forest Department. I wanted to thank the technical assistant, my sister Navamani, who helped me overnight in terms of analyzing all the sample. I should remember her actually now. And then I thank my principal and the staff member from my college. I also thank the former professors, teachers who taught me about the research in good manners. I would also thank the director and scientist who has worked the past and then working in the present in SACON. At last, I wanted to thank my beloved friends like my field assistant, lab assistant, who used to always with me whenever I suffer or whenever I am happy. So with that, I would thank everybody. I also thank my teachers, my ethic teacher Salim Ali, who actually taught me about birds and other things. So I also thank him, and then I thank the BCF team for the opportunity given.